So welcome to the lecture of today about nociceptors and pain perception. I will introduce you to the topic, speaking about nociception and pain, how nociception works and how a nociceptor is defined, the characteristic of nerve fibers and dorsal root ganglion, how to test nociception, the different pain models that we use to test nociception, and the type of pain, and at the end as well the intracellular mechanisms that are involved in pain, and an overview on pain-specific syndromes and pain therapy. So what about if you have a needle and you just pinch your lip or your arm? The sensation is the same? Do you feel the same? Actually not. So the lips have more sensory neurons, as you can see in this draw, and your arm less. This is why you feel more on your lips. So the representation of your body parts is different in the somatosensory system. And this is nicely represented by the sensory homunculus from an idea of Dr. Painfield. So what is nociception? Nociception comes from nocere, injury, and is a physical action of exciting small caliber primary endings via different stimuli, like mechanical, thermal, or chemical stimuli. So your body detects these stimuli, or if there is a tissue damage, and protect yourself by this tissue damage through nociception, through different signaling. However, you feel pain, right, sometimes. So pain comes from point, penalty, and is defined as the unpleasant sensory experience that normally is associated with tissue damage. However, there can be also pain without tissue damage. When we speak about nociception, we should think about how the pain perception is. So if you have tissue damage, the response of your sensory neurons, so your nociceptors, could be increased. You could have an increase of pain sensation. And this is called hypersensitivity. So when we speak about nociception and pain perception, we have to think about two important words, allodynia and hyperalgesia. Allodynia is the pain resulting from normally painless stimuli, and hyperalgesia is an excessive response and sense of pain to a normally painful stimuli. So, what is a nociceptor? And how a nociceptor works? A nociceptor is a free nerve endings in the skin, which is stimulated by different stimuli, like mechanical, thermal, or chemical stimuli. Whenever a stimuli comes and excites the nociceptor, or a tissue damage excites the nociceptor, you have different pathways activated. So what happened? There is a change in the membrane potential of your nociceptor that is called receptor potential. This is then encoded in a series of action potential which travel along the axon of your nociceptor and reach then through the spinal cord the higher side, the anatomical side of your body. So where are the nociceptors located? As you can see here in the picture on the left, as I told you, nociceptor is a free nerve ending in the skin that feel the stimuli, different stimuli. However, the body of the nociceptors are located in these structures here that are called dorsal root ganglion. And then they synapse, with in the dorsal horn of the spinal cord, in a specific lamina of the dorsal horn of the spinal cord. As you can see, these dorsal root ganglia are located along your body, but actually they don't innervate organs that are located on the same level of your body. But for example, as you can see here for the lumbar level, we have L1 and L2 dorsal root ganglion neurons, or afferents, that innervate parts that are in the lower part of your body, like the urinary bladder or the lower part of your body, like for example the upper limb. So to have an overview, we can say that the nociceptors, so the free nerve endings, are located in the skin. Then the body of the nociceptors are located in the dorsal root ganglion. And these then synapse with neurons in the dorsal horn of the spinal cord. So the signal is generated actually in the periphery, at the nerve terminals. Then, as I told you before, there is the receptor potential that is encoded in a series of action potential, which travel along the axons, and then at the end, through pathways, reach the higher structure of your brain. So actually, there are different types of nerve fibers, as you can see here, that then synapse with neurons in the dorsal horn. So the different fibers are classified by their diameters. We have large myelinated fibers and small unmyelinated fibers. In the first class, we find A alpha, A beta, and A delta fibers. As you can see, they have a diameter that varies from 17 micrometer to 1 to 5 micrometer. And in the second class, we have C fibers, so the unmyelinated small fibers, because as you can see here, the diameter varied from 0.2 to 1.5 micrometer. This then, it's important for their conduction velocity. So, bigger are the fibers and myelinated are, then you have a faster conduction velocity. While the C fibers, as you can see here, they have a slower conduction velocity, from 0.5 to 2 meters per second. This means that also when a signal comes, the first fibers that are interested to generate the signal are the A fibers, A beta and A delta fibers for nociception more. So the first signal that comes to your brain is this kind of sharp pricking sensation. That it's a first phase that comes from the conduction velocity, from the conduction of these fibers. While the second feeling, that is more a burning sensation, comes from the signaling from the C fibers. So these fibers, as you can see here, they synapse the A, beta, and delta fibers with neurons in lamina 1 and the outer laminar 2. And the C fibers, they synapse in neuron with the inner laminar 2. So there are also different receptor types. So these fibers react to different stimuli. For example, A delta fibers, there can be A delta fibers that react to mechanical stimuli only, A delta fibers that react to thermal and mechanothermal stimuli, and then the C fibers are defined as polymodal. So they react to mechanical, chemical, and thermal stimuli. These are the different responses I was telling you before. These fibers are characterized actually by the expression of different channels in the plasma membrane. For example, the nociceptors that respond to heat, they express TRPV1 channel, for example, that is a channel of the transient receptor potential family. The nociceptors that react to cold, they have TRPM8, that is another channel that from the same family. And the polymodal fibers, they have a variety of channels and molecules expressed in their plasma membrane. So, as I told you before, dorsal root ganglia, they are the structure where the cell body of your nociceptor is located. You can distinguish different populations in dorsal root ganglion. So they differ by the expression of different molecular partner, like the isolectin beta-4, IB4, the calcium gene-related peptide, CGRP, and neurofilament 200 and F200. These two populations are important, the IB4 and the CGRP, for the nociception. So as you can see, you can stain your neurons in the ganglia with CGRP and IB4, and you can find different populations. One is the peptidergic, and the other one is the non-peptidergic. We will see why this is important afterwards. You can find the same staining also in the dorsal horn of the spinal cord. So how you can test nociception? There are different techniques. For example, in vitro, you can record the electrical signal coming from your nociceptor. So if you have DRG neurons, dorsal root ganglia neurons in culture, and you stimulate with different substances that activate your nociceptors, you can then record the electric signal that comes from your nociceptors. For example, you can also understand which type of fibers are involved, A beta, A delta, or C fibers. You can do a skin nerve recordings and understand the conduction velocity upon stimuli, for example, mechanical stimuli with von Frey filament. And you can record this electric signal and distinguish between different fibers. In vivo, we have different tests, like von Frey test for mechanical stimuli. You have a precise filament with a standard, with a precise, a defined force, which can stimulate the paw, for example, of a rodent. Upon this stimuli, you can calculate exactly the mechanical threshold when these rodents can react to the stimuli. The same is true for the Hargrave test, which is a test where you stimulate the paw of the rodent with a heat stimuli here. Then you can understand and analyze the heat threshold. As well for human, we find a variety of tests that are similar to the one I showed you before. For example, the von Frey is also found in tests that are used for patients. This is the pinprick test, so you have the same kind of mechanical stimuli. Then you can have a pressure stimuli in the QST pressure test, and you can measure this threshold and other type of stimulation. Upon stimulation, it was possible to see that if you stimulate different parts of your body, this corresponds to parts activated in your somatosensory cortex, and you can see this with magnetic resonance imaging. Other type of stimulation, there are also thermal stimulation, for example here, as you can see, and chemical stimulation with compound like capsaicin, that is the compound of the hot chili peppers, or mustard oil. So these studies in vitro and then in vivo, or these studies in vitro and then on animal models, that I will show you in the next slide, are important as a translational bridge for human studies. So as I told you, there are different pain models that resemble the type of pain. So, for example, we can see the spare nerve injury, where the common and the tibial nerve are cutted, and only the sural nerve is intact. Or the crash, where there is a crash is performed on the sciatic nerve. The chronic constriction injury, where you have 4 ligatures on the sciatic nerve. And then the SNL, where you have a ligature here on the nerve. Other type of models are, for example, the one that resemble an inflammatory pain, where you inject in the paw of the rodent a compound, like, for example, the CFA, the Freud adjuvant compound, which stimulate an inflammatory damage. So with these pain models, you can study pain sensation and nociceptions. And this is why it's important also to perform tests as well in models. So we touched the first topics. So what is nociception? What is pain? How the nociception works? What is a nociceptor? So remember, the nociceptor is a free nerve endings in your skin that actually is activated upon stimuli, as we say. There are different fibers that are involved in this, how to test nociception, and as well the pain models that are available nowadays. So we will go through now the pain pathways that are involved in nociception and pain perception, and then we will have an overview on some pain syndromes and pain therapies. So what happens upon, for example, tissue damage? Tissue damage, as you can see here, activate different cells. And then you have release of chemokine, cytokines, for example, the one from the interleukin-6 family, that are important in pain sensation and pain signal transduction, or growth factors like NGF, nerve growth factors, that bind their own receptors on the plasma membrane of the nociceptors, and then the signal is transduced through the axon of the nociceptor. So, for example, NGF, nerve growth factor, is a really important factor because it's actually involved in the development of the nociceptors. So nociceptors respond to NGF because they express TRKA receptors. However, some nociceptors, during their development, they lose this receptor and they start to express another type of receptor, called RET. This is the receptor for GDNF and other factors and other growth factors, which bind its own receptor, and it's important as well for pain signaling. So as you can see here, then, we can recall our two classes of nociceptive neurons, the CGRP positive neurons, so the one that they express the peptide, so the peptidergic population, and the IB4 positive neurons, so the one that they express isolectin beta-4, the non-peptidergic population. You can see that actually they also are divided because they respond, one to NGF and the other one to GDNF. As I told you then, the fibers, the nerve fibers that respond, for example, to this factor, to this growth factor, then travel along and synapse with neurons in the lamina 1. So for the CGRP positive neurons in the lamina 1 and lamina 2 in the outer layer, and for the IB4, for example, for C fibers, if they are C fibers, they synapse in the inner layer of the lamina 2. Another type of molecules, a partner that is involved in pain sensation, is the TRP channel family. So as I told you before, this transient receptor potential channel family, it's important in the pain sensation because it's able to be activated upon different stimuli, for example, heat stimuli. This is the case of the TRPV1, one of the members, that is activated at 43 degrees. So this channel is also activated by capsaicin, as I told you before, the component of the hot chili peppers, and is important and is expressed on the nociceptors that respond to heat. Other type of channels are, for example, TRPM8 that respond to cold stimuli, or the family of the ankyrin one, TRPA channels, in particular TRPA1 channel that is involved in mechanical stimuli. So as you can see, heat stimuli can activate different channels, TRPV1, 2, 3, or 4, with different temperature range, or cold stimuli that can activate A delta fibers, for example, activating TRPM8 or TRPA1 channel, and the mechanical stimuli, as I told you, the channels, for example, the TRPV2 channel or TRPA1 channel. In the class of channels that are important for pain sensation and nociception, we also find voltage-gated sodium channels and voltage-gated potassium channels. Because these are really important channels, because actually nociceptors predominantly express NaV 1.8 channel, so they can be recognized by this. And why these channels are really important? Because, for example, if you have change mutation in NaV 1.8 or NaV 1.9 gene, you can have human pain disorders. For example, patients with loss of function mutation of these channels are unable to detect noxious stimuli, and suffered as of severe injuries. Gain of function mutation of another channel of the same family, the NRV1.7, leads, on the other hand, to hyperexcitability and to extreme pain disorders, like, for example, erythromelalgia. So, but these are not only the molecular patterns that are involved in pain sensation and nociception, but as well there are neurotransmitters that are involved in this. So which are the most important neurotransmitters that are involved in pain pathways? Substan P and glutamate are really important because actually upon stimuli of the first order nociceptive neurons, there is a release in the synapses from these neurotransmitters. These neurotransmitters then stimulate second-order spinothalamic neurons, which cross contralaterally and then form the neospinothalamic tract. In this way, the signal is then able to reach higher structure on your brain, like thalamus and then the somatosensory cortex. Obviously, there are also inhibitory mechanisms involved. For example, interneurons, which release enkephalin, are involved in the inhibitory signal, because enkephalin can reduce the calcium release, and then there is a decrease as well in the neurotransmitter release. So it's a control system. So substan P, that is this important neurotransmitter involved in nociception, belongs to the family of tachykinins. It's a peptide of 11 amino acid, and it binds its own receptor, the neurokinin 1 receptor, that is actually a G protein coupled receptor. It's localized in the synaptic vesicles, so in the nerve endings, and then is released, as I told you, upon stimuli. So it's released from the nerve endings in the dorsal horn of the spinal cord and stimulate the second order nociceptive neurons. So this neurotransmitter is involved in nociception, but also in vasodilatation, for example, in the periphery, and in the wound healing. So now I will give you an overview as well, a short overview, on the different pathways that actually modulate the pain sensory mechanism. So more in general, we can say that there are two pathways. One is called ascending pathway, and the other one is the descending pathways. So the important one, the ascending pathway, is the one that we know. So upon stimuli, you have actually the excitation of your nociceptors, then the stimulus travel along, there is the synapses in the dorsal horn of the spinal cord, then, as I told you, there is a cross of the contralateral to the contralateral side, and this goes along up onto the thalamus and then to the somatosensory cortex. So this one is called neospinothalamic tract. On the other hand, for example, you heard about soldiers that coming back from war and they have severe injuries, but they don't feel pain. This is due to the action of the descending pathways, that is actually an inhibitory pathway, and is gone from higher structure of your brain to the dorsal horn of the spinal cord. So to conclude, I also want to show you some of the pain syndromes. So as I told you before, there are different types of pains. The physiological pain that is related to tissue damage, the inflammatory pain that is more mediated by immunological, the immunological system, neuropathic pain that can be associated with the first two. So as I told you before, pain is a reaction of your body to protect yourself. So it's a protective mechanism. But sometimes this protective mechanism is not really normal. For example, this is the case of the chronic pain, that there is no helpful function when there is chronic pain. One type of chronic pain is the neuropathic pain. And this happens when there is a lesion or a damage on your nerve, for example, or to the primary nociceptors. Other type of specific pain syndromes are, for example, the hyperalgesia, where there is an enhancement of the sensation of pain, which results from tissue damage. And there are different substances involved in this, also substance P. Or the causalgia, where you have this burning sensation by increased sympathetic efference activity. Or the neuralgia, where there is this kind of severe persistent pain in the cranial or in the spinal nerve. The thalamic pain syndrome, or the referred pain, for example, where there is pain arising from deep visceral structure, and then it's felt on the surface of your body. And the phantom pain, where that is due to an overactivity of the dorsal or neurons. So it's this false feeling, for example. So there are obviously pain therapies which help to improve the quality of the life of patients. And these are really important. But to reach to this point, obviously, it was important to study nociception and pain perception. There are different approaches, as you can see here in the slide. So physical approach, like the rehabilitation or the new light therapy, or psychological approaches with the cognitive behavioral therapy, for example. And then apomedication, as we know, for example, opioids, analgesics, or non-opioid analgesics. Antidepressant drugs, they are also used today for pain therapy. And at the end, interventional procedure, so surgical procedure. All these therapies, all these different approaches, they are then able to improve the quality of the life of patients. So in conclusion, we can say that nociception and pain perception, it's important for the survival and the protection of your body. Thank you very much for your attention.